Good morning, it's August 29th and this is your daily brief in science. Here's everything you need to know. A recent study from McGill University has unveiled a crucial discovery in the realm of neuroscience. Researchers have pinpointed a small group of dopamine neurons in the striatum that play a vital role in balancing brain functions associated with reward, cognition, and movement. This significant research, led by Bruno Giros, is the result of nearly a decade of collaboration with the Université Libre de Bruxelles, drawing on Giros' extensive 30 years of experience in the field. By employing innovative genetic tools, the team focused on dopamine receptors that represent only 5% of the dopamine neurons in the striatum. Their study identified a previously uncharacterized group of dopamine receptors that integrate the functions of both D1 and D2 receptors, which are essential for maintaining dopamine balance in the brain. Giros highlighted that these neurons are critical for achieving equilibrium between the activating and inhibiting effects of dopamine, a key factor for normal brain function. The newly discovered dopamine pathway is pivotal in regulating dopamine levels, preventing overactivity that can lead to disorders such as schizophrenia, addiction, and Parkinson's disease. The implications of these findings could lead to new treatment avenues for various psychiatric disorders, including schizophrenia, ADHD, and addiction, as well as neurological conditions like Parkinson's and Alzheimer's. Looking ahead, the research team plans to investigate how this dopamine pathway influences cognitive processes and its potential implications for psychiatric disorders. The findings of this study were published in the journal Nature Communications, representing a significant advancement in understanding dopamine pathways and their impact on brain functions. Funding for this research was provided by the Canadian Institute for Health Research and the Graham Beck Foundation. This research not only addresses the serious issues stemming from dopamine imbalances but may also offer insights into potential treatments for anorexia nervosa. A recent study published in the Journal of the American Medical Association reveals a significant decrease in the risk of death for kidney donors over the past decade. This analysis examined data from 164,593 kidney donors between 1993 and 2022, focusing specifically on mortality rates within 90 days post-surgery. Researchers divided the data into three eras: 1993 to 2002, 2003 to 2012, and 2013 to 2022, highlighting advancements in donor selection and surgical techniques. By 2022, the findings indicated that fewer than one death occurred for every 10,000 donations, with only 36 deaths recorded over the 30-year period. Mortality rates for kidney donors have dropped from 13 deaths per 10,000 during the 1993 to 2002 period to just 5 deaths per 10,000 from 2013 to 2022. Lead author Alan Massey, Ph.D., emphasized that kidney donation is now safer than ever, with mortality among donors being extremely rare. The evolution of surgical techniques has played a crucial role in this decline, with laparoscopic nephrectomy now considered the standard procedure, significantly reducing complications. Dr. Dori Segev, a transplant surgeon at NYU Langone Health, noted that it is increasingly safe to donate a kidney. The study also advocates for updating current guidelines for donor-informed consent, which are based on outdated data from 2009. Healthcare professionals are optimistic that this reassuring data will encourage more individuals to consider becoming living kidney donors. With nearly 90,000 people currently waiting for a kidney transplant in the U.S., living donations can significantly reduce waiting times. However, the study also highlighted that male donors and those with a history of hypertension face higher mortality risks, underscoring the need for careful donor selection. Researchers at the Sainsbury Wellcome Center at UCL have made significant strides in understanding how specific groups of neurons interact to mediate sensory prediction error signals. The study focuses on V1P-expressing inhibitory interneurons and the pulvinar, both of which play crucial roles in processing unexpected sensory events. When activated, VIP neurons can either suppress or enhance activity in the neocortex, facilitating the detection of these unexpected stimuli. The pulvinar is essential in amplifying sensory responses in the neocortex, particularly in response to unexpected stimuli. Neural recordings from the study showed that unexpected stimuli elicit stronger responses than expected ones, indicating the presence of prediction error signals. Further experiments revealed that these signals do not just encode the difference between expected and actual stimuli, they amplify the representation of unexpected sensory input. To explore these dynamics, the researchers utilized a virtual reality environment where head-fixed mice formed predictions about visual stimuli presented at specific locations. The study underscored the importance of a specific thalamocortical disinhibitory circuit in generating sensory prediction error signals in the primary visual cortex. Professor Sonia Hofer highlighted the significance of understanding these neural circuits to improve learning from mistakes. The findings may have implications for understanding alterations in neural circuits associated with disorders such as autism spectrum disorders and schizophrenia spectrum disorders. Future research will further investigate how the brain compares predictions with actual sensory inputs and how these processes drive learning. This collaboration between the pulvinar and VIP interneurons is crucial for amplifying unexpected sensory input, generating essential prediction error signals for learning and adaptation. Scientists are sounding the alarm about the potential disappearance of the Y chromosome in the next few million years, a development that could lead to human extinction unless a new sex-determining gene emerges. In humans, males have one X and one Y chromosome, or XY, while females have two X chromosomes, or XX. The Y chromosome is currently home to approximately 55 genes, a stark contrast to the 900 genes found on the X chromosome. One critical gene located on the Y chromosome is the SRY gene, which is essential for the development of male reproductive organs. This gene activates around 12 weeks post-conception by stimulating the SOX9 gene. However, over millions of years, the Y chromosome has experienced a significant loss, shedding 1,393 of its original 1,438 genes, as noted by genetics professor Jennifer A. Marshall-Graves. Professor Graves highlights that this gene loss is part of a broader historical trend regarding sex chromosomes. Research led by Asato Kuroiwa has revealed that some rodent species, such as spiny rats, have adapted by relocating genes from the Y chromosome. In these cases, a DNA duplication near the SOX9 gene on chromosome 3 has compensated for the absence of the SRY gene. This study points out that two rodent lineages, specifically mole voles and spiny rats, have successfully developed alternative mechanisms for male sex determination after losing their Y chromosomes. Future research will explore these genetic changes further, aiming to shed light on sex determination and the implications for the survival of male offspring. If the trend of Y chromosome loss continues, there is a risk that only female offspring could be born, raising significant concerns about the long-term survival of the human species. Should humans evolve separate sex determination systems, future visitors to Earth might encounter multiple human species with varying sex chromosomes instead of a single type. Tuberculosis, or TB, continues to be the leading cause of death from infectious diseases globally, affecting millions. A recent study published in Nature has shed light on the critical role of the immune signaling protein, TNF, in inflammatory responses, and its narrower function in immunity than previously understood. Conducted by researchers Stéphanie Boisson-Dupuis and Jean-Laurent Casanova, the study highlights the genetic basis of recurrent TB linked to TNF deficiency. It focuses on two patients from a consanguineous Colombian family who suffered from severe recurring TB infections despite having normal immune systems. Genetic analysis revealed that both patients had a mutation in the TNF gene, which is essential for regulating immune responses and inflammation. Whole exome sequencing identified a frameshift variant in the TNF gene that was common to both patients, suggesting a genetic cause for their TB. The absence of functional TNF resulted in an inability to produce reactive oxygen species, or ROS, which allowed Mycobacterium tuberculosis to proliferate unchecked in the patient's lungs. This impaired ROS production in macrophages is crucial for controlling TB, underscoring TNF signaling's importance in macrophage function. These findings challenge traditional views on TNF's role in immunity, particularly its connection to TB susceptibility. The research also explains why TNF inhibitors, often used to treat autoimmune diseases, increase the risk of TB by compromising immune defenses. Ultimately, this study suggests a re-evaluation of TNF's role in immunity, paving the way for new treatment strategies that focus on other cytokines responsible for immune responses. With about 25% of the global population infected with MTB, understanding genetic factors is vital, as only 5 to 10% of those infected will develop the disease. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.